Cell biology. Alright, you've got only one part of one chapter on cells. It's not enough. But I'll tell you, there's two major kinds of cells on this earth. And the two major groups are prokaryotes and eukaryotes. And you might think of one as old and one as new. I don't care, but the reality is we're both here. And our cell structures are very different. And we both count on each other for our survival. Well, gosh, at least we're counting on the prokaryotes. So prokaryotes are simpler cells in the sense that they're smaller than the eukaryotic cells. And they are, prokaryote is thought to is interpreted directly as before nucleus, right? But they don't have a true nucleus. And they are things like bacteria and archaea. Those are the things that are these prokaryotes. They live in, they're extremophiles that live in hot springs. They are also bacteria that live in your gut, and they are also bacteria that live in your bathtub, wherever you might find them, or in your old chewing gum. These are organisms that are not unsophisticated, they just are smaller. Their molecular machinery is pretty complicated in a lot of ways, and so we probably shouldn't look down on them too readily as being lesser organisms, but they are very different. Now let's talk about eukaryotes. Prokaryotes are the little ones. Eukaryotes are a lot bigger. I mean, if I were to compare a prokaryote to a eukaryote, gosh, I think if I were to say this is a prokaryotic cell, then a eukaryotic cell would be at least this big on average. Okay, I'm holding up my big textbook from current issues here. What do we got? Human biology. So, eukaryotes are a lot bigger. What this would mean in your class, if you were here, is that you could see them on a microscope, the eukaryotes, but you can't really see prokaryotes well because they're too small to see in a standard microscope. You have to have a really, really nice microscope. So, those are two big differences. The two big differences. Eukaryotes have a nucleus, and that just means a nuclear membrane holds the DNA into one area of the cell. And it's bigger. There you go. Now, we are eukaryotic organisms. We're multicellular eukaryotic organisms. And you can go through the kingdoms lecture, I guess, was in chapter 1 in there, and so you can go and look at all those differences. But what I want to talk about when I do the cell, we're looking at the eukaryotic cell primarily, and doing a tour of that cell. Now, this is an easy section of the book. All you have to do is look at the different organelles and see what they do. You may, if you like, visualize the cell as a car motor. How it functions is has these different components in there, and distributors and crankshafts and whatever you want to look at. I don't know much about cars. So, but you can also look at it as a cell. So let's talk about some of the major organelles that are in there. And the first group I'm going to talk about are what are called part of what's called the endomembrane system. And that means that the tissues that make up the membranes that bind these things together are all very similar structure. They're all made of a phospholipid membrane that has a commonality with this group. And there are a couple of organelles, mitochondria and chloroplasts, that don't fit the mold. They're very different. We think they have very different origins, but they are outside the endomembrane system. Number 1, nucleus contains the DNA. That's about it. It's just an envelope wrapped around the DNA. All right? See on my list here, rough endoplasmic reticulum. Now, also known as the rough ER. The rough part comes from ribosomes that are on this sort of endomembrane membranous cloud that's out there that makes a space inside of it, and inside that space protein folding is occurring. So if you want to make proteins, proteins are made by the rough ER. If you make secretions of proteins like digestive enzymes, they're made in the rough ER and then they're sent outside the cell. Right? That was easy. Smooth ER is different from rough ER. It's part of the same long membrane thing, but it doesn't have the ribosomes on there that do this making of the proteins. So we call it smooth ER, and its primary function is detoxification. There's some other things that it does. let's go with detoxification for now. What have I got? The ribosomes themselves are these structures that are on the rough ER. There are some free-living ribosomes as well, and they actually do the assembly of proteins. So they read messenger RNA and they translate it into protein sequences, like a sequence of amino acids that makes the protein. All right. Next one is Golgi Complex. Very strange little organelle. It just sort of packages things that come off the rough ER into a transport vesicle and then sends them off to where they need to go. They do some modifications as well, but they call it the shipping center. But essentially, if you're a protein being made in the rough ER, to get out of the cell, you're going to have to go through the Golgi complex and get packaged and then sent somewhere so you don't get lost in the cell en route. All right. Now the lysosome is a really nice organelle because the lysosome is a garbage disposal. Essentially, you have all these enzymes in there that cut stuff up. And so in this lysosome is where you take some old molecule or some old piece of junk you're not using anymore in the cell, take it into the lysosome, and it gets ground down, cut down into basically its smallest pieces. And then those pieces can be recycled or eventually get excreted as waste somewhere. There's a nice example of a disease in the written lecture that I gave you that is a defect in one of the enzymes in the lysosome. It's a tragic disease, but it's a very interesting example of just how important each organelle is. Transport vesicles are vesicles that they're just pieces of like endomembrane system, right? This phospholipid membrane that carry collections of proteins and different structures around in the cell. So if you're going to send some protein, like I said, to go out and digest something, you make it in the rough ER, you send it to the Golgi, and then what buds off of the Golgi are these vesicles that travel to where those proteins need to go. That's it. I mean, that was pretty straightforward. Prepare yourself for an exam question that deals with these. There's a good quiz on there. Now I'm going to remember that mitochondria and chloroplasts are very distinct from the others. It is thought that these were once prokaryotic organisms that were ingested by an ancestral cell because they act just like bacteria. They have their own DNA inside them. They divide inside your cells like bacteria do. The DNA looks like bacterial DNA. It's pretty cool. So check it out in my written lecture. Look at some of that. But what's neat about them is they do important things, like mitochondria does. Take sugar, like that glucose molecule, and turns it into ATP, which is an energy molecule. And it can do some work in your cell, like power enzymes or power muscle function, for example. So remember, mitochondria take sugar and it makes energy in the form of ATP. And chloroplasts are going to be in plants, right? Not in humans. But plants also do have mitochondria. They get both of them, mitochondria and chloroplast. And the chloroplast takes sunlight and makes it into energy. So it's a fantastic process that we don't have time to go into in this class. But essentially, from the light of the sun and some oxygen, some carbon dioxide from the air, not oxygen, sorry, some carbon dioxide from the air and some water, this thing makes sugars. And that is the foundation for the vast majority of energy on our planet that is accessible to humans, is the fact that chloroplasts can make sugars out of sunlight, essentially. It's pretty amazing. So there you go. Those are the cells. That's the highlights of the cell chapter. Hope it's been a good 5 or 6 minutes.